The Cell Cycle by Brianna Harris. The cell cycle is the life of a cell from the time it is first formed from a dividing parent cell until its own division into two cells. All eukaryotic organisms have a characteristic number of chromosomes in their cell nuclei. Somatic cells, which are body cells in form of main tissues, have 46 chromosomes, which is the diploid chromosome number. Mitosis is the process by which somatic cells divide, forming daughter cells that contain the same chromosome number as the parent cell. This is also asexual reproduction. Gametes, known as sex cells, sperm and egg cells, are haploid and have half the number of chromosomes as a diploid cell. Human gametes have 23 chromosomes, and they are formed by the cell division called meiosis. A genome is known for carrying the genetic information in a cell. Mitosis's first phase is called interphase. Interphase is also the longest phase of mitosis and is 90% of the cell cycle. The first checkpoint in interphase is called G1 phase. In G1 phase, the cell grows while carrying out the cell functions that are unique to the function and structure of that cell type. In checkpoint 2, called the S phase, also known as DNA synthesis, the cell continues to carry out its unique function, but does one important process. This is the only phase where the chromosomes are duplicated, which means if there were 6 cells in G1, there are now going to be 12 cells in phase 2, phase S. The G2 phase is where the chromosomes that have been duplicated arrive just before mitosis. Mitosis can be broken down into interphase, prophase, and prometaphase. In prophase, the chromosomes become more tightly coiled into discrete chromosomes. The nuclei disappears, and the mitotic spindle begins to form in the cytoplasm. As you can see in interphase, here's our nuclei, and we have the nuclear envelope. And in prophase, the chromatins and chromosomes begin to separate and condense. Also, the nuclei has disappeared and it is no longer here. The mitotic spindles begin to form as well. In prometaphase, the nuclear envelope begins to fragment. So it begins to break apart, which allows the microtubules to attach to the chromosomes. And as you can see, here are the microtubules, and they are attaching to the chromosomes inside the dismembered nuclear envelope. The two chromatids of each chromosome are held together by protein connectors in the centromere. The microtubules will attach to the connectors. In metaphase, the microtubules move the chromosomes to the metaphase plate, which is also known as the equator of the cell. The microtubules are now referred as spindle. The centrioles have migrated to opposite poles in the cell and are developing on the spindle. In anaphase, the sister chromatids begin to separate. They are pulled apart by motor molecules interacting with connector microtubules. The cells elongate as the non-connector microtubules separate, again with the help of motor molecules. By the end of this phase, the opposite ends of both cells contain complete and equal sets of chromosomes. Telophase. Telophase is the phase where the nuclear envelope reforms around the sets of chromosomes located at opposite ends of the cells. So as you can see from the other picture, the nuclear envelopes have begun to form around the chromosomes, and it's beginning to split off into two new cells with equal number of chromosomes on each cell. The chromatin fiber of the chromosomes becomes less condensed. Another part of telophase is called cytokinesis. Cytokinesis begins when the cytoplasm of the cell is divided. In animal cells, cleavage furrow forms and eventually divides the cytoplasm. In plant cells, a cell plate forms that divides the cytoplasm. Meiosis involves gametes, which are sex cells. Meiosis starts with homologous pairs, which are chromosomes with the same characteristics, for example, length or gene. The two homologous pairs then go through synapsis. Synapsis is the fusion of chromosome pairs. After synapsis, the homologous pair goes through crossing over, which is also known as recombination. Crossing over, or recombination, is the process in which homologous pairs exchange parts of their DNA with each other. After crossing over occurs, they go through meiosis 1. Meiosis 1 is the first division in meiosis. This is where the recombinant chromosomes separate into two. In meiosis 2, this is where the sister chromatids separate into 4 haploid cells. So each cell contains 23 chromosomes. So this contains 23 and this contains 23. Each one gets half of the chromosomes from each parent. So this could be the mother, this could be the father. When you add the 23 chromosomes from each set together, you'll end up with the 46. Meiosis, the good thing about meiosis is that it provides genetic variation because you get 2 different sets of genes that you can inherit from either parent. Genetic variation is good because if you have the same set of genes throughout an environment, if one environmental action like a disease spread around, then all organisms would die off because they had the same genes and same inheritance. The eukaryotic cell cycle is regulated by a molecular control system. The most common regulators are cyclin, MPF, PDGF, and CDKS. The steps of the cycle are controlled by a cell cycle control system. This system is what helps move the stages from G1, G2 to mitosis. The G1 phase checkpoint is the most important, which is also controlled by cyclin. If the G1 phase is able to go ahead throughout the process, then usually the whole cell cycle will occur. As you can see here, the cyclin concentration is always highest when starting mitosis because cyclin is necessary for the process of mitosis to start. Without cyclin, the mitosis process wouldn't start and neither would cell division. MPF is the regulator that passes on to the G2 checkpoint, which initiates the events of mitosis. During anaphase, MPF switches itself off, so by starting a process that leads to the destruction of the cyclin molecules. If the cyclin molecules are not able to function properly, then it brings a close to mitosis as a whole, because cyclin is what controls mitosis. So without the MPF, it brings it to a close. Disruptions in the cell cycle include the gene P53, which is a tumor suppressor gene. The P53 protein can activate the P21 gene, whose product halts the cell cycle by binding to a cyclin-dependent kinase, which allows time for DNA to be repaired before cell division reoccurs. The P53 protein activates a group which inhibits the cell cycle and turns on genes directly involved in DNA repair. The DNA damage is too great to repair, so the P53 protein activates suicide genes, which is also known as apoptosis. Apoptosis is basically when the cell just kills itself. So in a normal cell division, it occurs slowly, but in cancer cell division, it takes over the cell and the cells begin to overproduce and overcreate.